welcome everyone. Thank you for giving up a couple hours on a Saturday on Mother's Day weekend. To all the mothers, happy Mother's Day early. And to those of you who have mothers, just a reminder, get them something tomorrow. This is the House Institute's Hearing Science Accelerator project. I'd like to welcome all of you here. This seminar today is on perilymph sampling, and so we want to explore the potential development of perilymph sampling protocols and how this could help us understand sudden sensorineural hearing loss. The seminar today, we're going to walk through a couple of different areas. We'll start by talking about our objectives and the agenda, and then provide an overview of the sudden sensorineural hearing loss diagnosis and hearing science accelerator initiative. And then from there, we'll dive into some research questions about perilymph sampling. I'm really happy that we've got four esteemed colleagues that are going to give us an overview of perilymph sampling, their experience of perilymph sampling from animal studies to human studies, and then also how this has actually made an impact on clinical treatment, which is, I think, pretty exciting. From there, we'll so the idea is the first hour we have focused speakers. The second hour, we will then have discussion, and then we'll kind of wrap it up at the end of that second hour. And the goal here is to help develop collaboration between just labs and also collaboration of thought. So a little different concept. We're going to try this format and see how it works. Hopefully this will work well, and we're going to do this with some other areas that will help kind of bring some different individuals together. We will be recording the meeting. We're going to then, after it's done, we'll edit out all the discussion part and then just have summarization of the four speakers and then kind of our consensus of what we decided. And the rationale for that is we do want to record it so we can make sure we get everybody's ideas in, but at the same time, we don't want to publish that so people can feel free to communicate. And then we'll also talk a little bit about the format, or maybe send you out a survey afterwards about the format to see what everybody thought about this. As far as how it was put together as a concept of moving forward with other kind of research ideas. So let's see, shims are the slides. Start sharing. Okay. So basically the objectives and goals is to review perilymph sampling and to look at what the current state is and how this would apply for sudden sensorineural hearing loss, potentially develop a protocol that would help our understanding of pathophysiology. We want to focus on early diagnosis and how this could actually help us with again, understanding sudden sensorineural hearing loss. Goals of today is maybe just to help us kind of come up with a standardization of human perilymph sampling, look at maybe how we could develop a role of what standardization for animal perilymph sampling, address additional questions that everybody may come up with. And so those of you who have a lot of experience with this, I'm sure will speak into this. And then talk about what's the best ways to collaborate between institutions, and maybe even have some individuals that want to step up and say, hey, we'd love to move forward with this project or that project. So let me give you a little brief overview then about, obviously, I think most of us understand what sudden sensorineural hearing loss is. It is a rapid onset. The Academy guidelines define it as happening within 72 hours. I would say most patients know exactly when it happened, and it is defined as having 3 frequencies of greater than 30 decibel hearing loss. At this point in time, I think everyone agrees there's no identifiable cause of when we talk about idiopathic sudden sensorineural hearing loss. There are other causes of sudden hearing loss, but for the purpose of this discussion, we're focusing on specifically idiopathic sudden sensorineural hearing loss. Next slide. So the Hearing Science Accelerator was put together as a concept to help us identify ways that we can develop new treatment options for some of the diseases for which there's been very little understanding. And unfortunately, in otologies, there's too many of those diseases. So one of the first diseases we've chosen is sudden sensorineural hearing loss. We're also going to be working on Meniere's disease, vestibular schwannomas, and acoustic neuromas. Next slide. So it's a 3-part process. The first is where we go through a process where we call disease prioritization. We put together a steering committee where we talk about who should be involved. The idea is we want to bring in experts in hearing science, but also experts from outside of hearing science to come together. We prioritize the disease in the sense of what are the issues we need to look at with the disease. And for sudden idiopathic sensorineural hearing loss, we chose diagnosis, etiology, and treatment. We then have a disease workshop, which we come together for a weekend. And that's what we did. And we had a manuscript that has been circulated, hopefully, to everyone. And then we go through this disease initiation stage, where not only do we, it's one thing to have a workshop and say, here's what we should do. But unfortunately, too many times what happens is that's it. And then some people pick up parts or pieces, and we're trying to push that process forward. So looking at over the next couple of years, trying to identify and answer the questions that were brought up in this workshop that we put together. So out of that, we came up with then different questions. Next slide, Jims. And so we had these research priority questions. The manuscript is listed here. So if anybody's watching this video later, or those of you who are on there now, we can provide this to you. It was published in Otology Neurotology back in October. The bottom line is it came up with 8 questions to say this is how we can advance the science regarding sudden sensorineural hearing loss. One of the questions, and this is a specific question for today, is can a biobank or patient registry be developed in order to help determine the etiology of sudden sensorineural hearing loss? And how would specimens be able to help us with this? And perilymph testing was one of those. Thinking about, is there some way to look at biomarkers, and would a test of perilymph be helpful in aiding in either the diagnosis, etiology, or treatment of sudden sensorineural hearing loss? And then the next question was that, could perilymph obtained during cochlear implantation or potentially even acutely at the time of onset be helpful at being able to identify something that would help us understand either the etiology or be a predictive marker for patients with sudden sensorineural hearing loss? Obviously, there's some questions about ethics of this, but this is what the group brought up and said this is a priority, and we should also look at standardization of perilymph collection to figure out how to yield this for maximum yield to potentially develop it into a useful clinical test. Next slide. So as I mentioned, this seminar will be uploaded eventually. And again, the open discussion in the second hour will be recorded but not uploaded, because again, we want to make sure everybody does have freedom. We would ask you to put your questions in the chats, and what we'll do is once the speakers have completed the presentations, then we'll go through the questions. When all the speakers have talked about it, then that will help us lead into the discussion part. So please, as we go through, if there's questions that come up or suggestions or anything, put into the chat, and we'll address those during the discussion phase. Next slide. So our 4 speakers today we have is Heinrich Sturkers. He's going to talk about perilymph sampling to develop biomarkers for sudden sensorineural hearing loss. Adele Moti is going to speak about PEG perilymph sampling protocols, some of the work that she's been doing. Tina Stankovic is going to talk about perilymph sampling in humans and some of the work that she's done for cochlear implantations and also labyrinthectomy procedures. And then Andrea Van Butis will talk about autoimmune inner ear disease and an overview of some of her current work and how this perilymph sampling has been helpful. So I appreciate all the speakers agreeing to talk. The first person that will talk will be Heinrich. Heinrich completed his medical education at Albert Einstein with a distinction in otology research, and then did his residency at Montefiore. I can never say that right, sorry. And then did a fellowship at Mass Eye and Ear, and he's currently professor in the Department of Otolaryngology at the University of Kansas. And so, Heinrich, thank you for joining us and giving us this this talk and the work that you've done, kind of pioneer work on perilymph sampling. So thank you for talking, and thank you for the work that you've done.
Hi, thank you, Bill. I think this is a real exciting project. For those of you in the audience that are a little confused, my title was originally perilymph sampling from stapedectomy. I will talk about that, but I wanted to take a little different approach to that topic. Okay, so we all know this I'm actually citing one of Tina's paper, that there is a mismatch between what the audiogram tells us and what is present in the cochlea below in terms of cells that are surviving. And that difference probably should be reflected somehow in, I guess I got to click this each time, right? So should be reflected in something that we can sample. So the inner ear is the only organ where biopsy, which is the basis of all of classic medicine and pathology, is not possible, or really has not been possible up to this point. You can do brain biopsies, you can even do retinal biopsies. But in the inner ear, traditionally, we have not had a way of taking a look at what is going on in the inner ear during active disease. And as you all know, developing our knowledge of temporal bone pathology is based on specimens from patients that have passed away and donated their temporal bones and are often years and years and years past the active disease process. So basically, you get to a phenotype by going from genome to transcriptome to proteome to metabolome. And the metabolome is the result of what your proteins and enzymes are doing. And any of these can be sampled to produce a biomarker. Sorry, I keep on hitting my space bar here. Habit. So for hearing loss at this point, we at this point have very well developed genetic testing in terms of whole exome sequencing. Whole genome sequencing is coming online. And we've now had quite a bit of animal work on proteomic sampling. The group in Hanover has done extensive work on proteomic sampling in humans. There's some early stage research on metabolomic sampling. And for example, there's a group in France that have shown that in perilymph taken from cochlear implant patients, they could differentiate long deafened individuals from less deafened individuals based on their perilymph metabolome. So, this is an area ripe for research and something that we as clinicians should really have. The real motivation when I got into this, I was running a clinical trial for hair cell regeneration in patients with severe to profound hearing loss. And we had one responder out of the 18 patients, and they were severe to profound hearing loss patients. And it became pretty clear that we had no good way of actually rationally picking our patient population. So a resident of mine, Matt Shu, who's now on staff at WashU, got interested in this and actually suggested that we look at microRNAs as biomarkers because they are very robust. There's been a lot of work in the eye on microRNAs related to glaucoma pathology, and a lot of work in the central nervous system. So we wanted to take a look originally if we could use microRNAs, which modify gene expression as a biomarker. Our initial paper on this was just looking at 10 patients undergoing cochlear implantation, stapedectomy, and labyrinthectomy, and we used a pulled pipette, and really the stapedectomy patients were included so that we had a normal hearing control, so a non-sensorineural hearing loss control. And basically, when you do your stapedectomy, it has to be bone dry. I usually, once the stapy superstructure is out, put a little piece of gel foam with epinephrine on the anterior part of the oval window niche and wait a little bit, and then we suction it, and then put the capillary in place and laser right next to it. And then usually when a little bit of perilymph comes up, you can get a very clean 2 to 5 microliter sample. When we did this, quite a few years ago already, we used a gene array, which has now been replaced by next generation sequencing. But this is how we got going. And we basically could identify about 300 microRNAs that were common to all patients. And then as we built our patient numbers beyond that, tried to look at microRNAs that were different between the labyrinthectomy group, which was our Meniere's group, the otosclerosis group, which was our stapes sampling group, and our undefined sensorineural hearing loss, which was our cochlear implantation patients. We used machine learning to try to sort of get at some of the differentiation between this. And after building a group of about 20 patients, we to train some models. So we used supervised machine learning, and we came out with an ability to really perfectly differentiate perilymph from otosclerosis patients and from Meniere's patients and from sensorineural hearing loss patients. And there was some commonality between the Meniere's patients and the cochlear implant patients that you could relate to sensorineural hearing loss, but there was a distinct population of microRNAs that could be isolated that related to Meniere's disease. And actually what we ended up finding in the otosclerosis patients, there was a distinct group of microRNAs that related to genes controlling bone metabolism that we could find. But interestingly, those were not the ones that differentiated the otosclerosis patients from the other in the machine learning model. Now, machine learning, you never really sort of get a good answer why initially why they why the different models differentiate, but we could using different types of machine learning models find slightly different microRNAs that differentiated the different groups. How am I doing on time, Bill? No? Okay. He hasn't pulled me off the screen yet. So we could actually
Doing good.
Okay, then also amongst our cochlear implant population, we could subdivide those into patients with a pure tone average of better and worse than 80 decibels. So, some residual hearing or really no residual hearing. And based on those, really accurately classify residual hearing from non-hearing perilymph. And these were the 3 microRNAs that really helped differentiate that. So, so that's that's really what got us started. And at least in the otosclerosis patients, we can very, very clearly say there was no loss in sensorineural hearing from the sampling procedure. And it's really pretty doable when you have a fixed footplate and make just a small fenestration. So when the footplate's fixed, the little sampling capillary really stays in good position. Okay, so why? The next obvious question is why are there microRNAs and everything else floating around in the perilymph? So microRNAs can be released just bound to protein, but more commonly they are bound in small vesicles that are produced by all cells that are involved in signaling between individual cells. And the really interesting thing is, again, from the eye literature, you can find extracellular vesicles related to glaucoma, both in saliva and in the peripheral serum. And there is a paper out too showing that extracellular vesicles in neonatal saliva can predict and identify children with autism. So obviously these EVs are transported and sent all over the place, and they actually protect the microRNAs and other substances that are inside. We've also looked from our perilymph sampling. I have a collaborator who is a microfluidics chemist who has developed a way of capturing extracellular vesicles, and she developed this originally as a test for ovarian cancer, and it is a magnetic bead that can attach to extracellular vesicles that you can then capture in a microfluidics chip. And we were able to pull out extracellular vesicles from human perilymph samples, and then immunogold stain them with gold labeled antibodies to prove that these are actually extracellular vesicles. So we can capture those and then resolubilize them, and then take these purified extracellular vesicles and put them through another selection step where we use an antibody against myosin 7, the hair cell specific protein. And in 5 patients, how we that we tested, we could actually pull out extracellular vesicles that have myosin 7 in them, so that their origin was likely from hair cells, auditory or vestibular. And from these, I don't have the data on here yet, we've been actually able to isolate microRNAs. So you can thereby, and you could use different antibodies for different cell types, now have a way of capturing data on what is inside or what is the activity of a single type of cell within the cochlea. So this is, I think, pretty much where I wanted to sort of stop things. This is all my collaborators. This is May was at KU. She's in Florida now. She's the microfluidics queen. This is my collaborator from Hanover, who's done all the human perilymph sampling. And this is a group also in Austria that synthesizes and grows GMP EVs. Thank you very much.
Heinrich, thank you. That was wonderful. Wonderful, both introduction and overview, and congratulations on your work. So looking forward to talking about this a little bit more. Let's move next to Dr. Muladi, is from a PhD from the Department of Otolaryngology at the University of Pittsburgh in the Pittsburgh Hearing Research Center. Her background is material engineering, where she received her PhD at North Carolina State University. She's been doing quite a bit of work in developing drug delivery models in large animals, and so we'll be interested to hear about her work that she's been doing in the basic science world. So Adele, thank you for joining us.
Thank you so much for giving me the opportunity to talk today, and welcome everyone to this seminar. I'm gonna see, oh, okay. All right, so today, as you heard from William, I'm going to talk about the pig model and the perilymph sampling from pigs. I'm going to make the introduction very brief, as you heard a little bit from Heinrich, kind of overviewing all of this, that we have problem diagnosing the pathology of the sensorineural hearing loss. And currently we have no method that we can clinically test that. So some of the promising solutions are proteomic and microRNA screening. And recently, as Heinrich mentioned, the metabolomic is also being added to these solutions. And the other one, of course, is the kind of combined approach. So first you identify the protein, and then you take a step back, you go find the microRNA that regulate the up or down regulation of those proteins. Another interesting avenue, again, Heinrich kind of mentioned, is using the extracellular vesicles that are released in the perilymph. They have a lot of advantages to make our job much easier for genomic and proteomic and even metabolomic. And the reason is that they're very selective in their content, they're tissue specific, they're stable and travel. And they are used as an early indicator. So we are working in that area too, but today I'm going to mainly focus on the model and not much about the exosomes. So what are some of the current challenges of identifying biomarkers? The first one is that the human perilymph samples are very rare. Most of the samples that we have from human patients are the ones that are severely injured, or from cochlear implant patient, vestibular schwannomas, Meniere disease, and so on. This is, for example, this is a work by Tina's group that they looked at the cochlear implant vestibular schwannoma patient. They identified the common proteins between them. This is another work from Heinrich Lab, as you heard about it, from Meniere disease, when they consider the otosclerosis as the normal kind of perilymph, and they compare to that, which brings me to the next point. And the fact is that the normal human perilymph sample are even more rare, right? So we need a normal sample in order to compare our diseased patient to identify biomarker. This is the only work in literature that I found, again, from Tina's lab, where they consider the meningioma cases as their normal standard, and they compare the patient to them to identify the protein as biomarkers. If you look at the vestibular dysfunction, you can see that they're not clustered as beautifully as the normal patient, which brings me to my next point, and the fact that there are multiple causes for the phenotyping that we observe. For example, if you look at the sensorineural hearing loss, we cannot cluster all the cases together. We need to be more specific. What is causing that? And that makes it very difficult to identify biomarker. So we need a controlled model for a specific case of the sensorineural. And then from there, we can take a step back, go back to the sudden sensorineural hearing loss, and then say, okay, these can be categorized in this group. So the objectives for our group is to first develop an animal model when it has similar characteristics to the human perilymph. That makes our job much easier in making a damage model for a specific sensorineural cases. Then we can look at the biomarkers. And then we have a lot of normal perilymph samples, which the first step is how comparable it is to human, if you want to use that as our normal. And the second objective is using the exosomes, which again, I'm not going to talk about it so much today. Now, this is the work that has been done in the literature when they looked at different, basically proteome from the perilymph across different species. You can see there's only 10% overlap between human, guinea pig, and cat. Cat has more overlap. Unfortunately, we are not using, or fortunately, nowadays in our experiments. And from Tino's lab, I think they mentioned that there is 10% overlap as well between the mice and human proteome. So another important thing is that, again, coming back to the normal perilymph sample, here they are using cochlear implantation and they consider that as their normal, versus the animals have a typical normal hearing. So how fair that comparison is and how we can actually standardize it is a challenge, right? We are suggesting that the pig would be a great animal model in order to looking into their perilymph as a replica of the human. The pig genome is very similar to the genome size is very similar to human. If we look at the amino acid sequences of some of the common deafness genes, we can see that pig and human have more overlap than human and mice, which is good. And also they have similar size and physiology to humans. They've been used for a lot of other organs for transplant and so on. So that's why we want to use pigs. I'm going to show you more of similarities between the pig and human. So this is the inner ear size. This is mouse. This is human. I guess you can appreciate that the pig and human are more similar. They have similar low frequency hearing to human, more or less, better than other animal models. And then we have this method that we can image the whole inner ear in 3-dimensional, which is post-mortem. I wish it was in vivo, but that gives us a lot of information, both in a cellular level, the structure, and also in the volumetric data. So this is a whole inner ear in a pig, newborn pig sample. When we can extract, for example, the length of the basilar membrane. And here I'm showing you again, pig and human are very similar, almost identical. We also counted the number of inner and outer hair cells. Again, human and pigs are identical. These are all kind of hinting us that the physiology and function are very similar in this model to humans. We use the same technique. We measured the different volumes of inner ear compartment. Here, I'm going to focus on the scala tympani. The reason is because perilymph is in the scala tympani that we want to collect. Looking at across the species, human and pigs are much more similar than any other animal model, including monkey and cat, which is very important. So the volume of the perilymph that we collect from humans is between 1 to 5 microliter. At least that's what typically we do. Even the minimum collection, if you look at the mice, you can see the whole skeletal-tympanic volume is 0.3 microliter. So even if you want to collect the whole volume, that's going to have different indication. We're going to get more contamination from CSF. We're going to have more contamination from probably endolymph. We're going to have very different profiling if you want to use that as animal model. And that also we have some techniques, for example, specifically for exosome, that we have minimum of the volume when we can collect exosomes. And probably it's very difficult to work in an animal model like mouse. Talking about the contamination from CSF, it's very important to look at the aqueduct location, which is the port of entry from CSF to perilymph. So this is a human sample. This is the aqueduct, and this is the round window membrane. This is the narrowest part of the aqueduct. And if you look at the picture of the pig, we can see that actually the size is very similar. That's very good. That means if we have contamination from CSF, if any, it's going to be similar. And also here I'm showing you that the aqueduct in humans, most of the humans, is covered with the soft fibrous tissue. And we observe the same thing in pigs as well. So these are all good news for us. What's the next step is to actually look at the perilymph sample in pigs and compare it to the human and see how similar it is to normal, maybe meningioma cases that Tina reported. So first is how to collect the perilymph. This is the skull of the pigs. I put here for you, maybe you can agree with me that they have a little bit different skull than humans. The location of the, for example, mastoid bone is different. The ear canal is much, much longer, narrower, and tilted, which makes our job way more difficult to access the inner ear. But luckily, we have developed this technique where we can access the inner ear. We published this last year, this paper. Basically, we open the back of the bulla, we drill kind of a temporal bone along with the ear canal. We access the tympanic membrane. In order to access the round window membrane, we need to open up actually further, both down and to the right. The round window membrane is fully covered with bone in the domestic pig, so we have to expose it by drilling further. This is the round window membrane partially exposed. And this is a fully exposed round window membrane. We are using a 5 microliter capillary tube to collect the perilymph. As soon as we introduce basically the capillary tube, you can see the perilymph is filling up the tube. Now, what are the benefits of using capillary tube versus the needle? The needle creates a suction, so it might damage basically the sensory cells or at the tiniest structure in the inner ear. It might create a leakage between the endolymph and perilymph, so we might get some contamination from endolymph. It creates a negative pressure exactly on top of the aqueduct, so we might get more CSF contamination. Here we didn't pull our tubes, but in our future we plan to pull our capillary tube so that it will create much less puncture or entry point. Another important consideration is, are we damaging the hearing while we are doing the perilymph sampling? These are the auditory brainstem responses that we collect from the pigs before and after our surgery. This is not after perilymph sampling. But we are showing here that the threshold of hearing is not changing both in the click and the pure tone responses in pig by using the surgical technique. So our next step is to actually test and see what happens after we do the perilymph sampling. And as I said, we are still doing the proteomic, we are doing the microRNA profiling. And with that, I would like to thank you all for hearing and listening to this talk, and our funder, specifically AOS, that is supporting this part of the project that I showed you.
Adele, thank you very much. It was a very nice overview of both perilymph and the different animal models. So thank you. And again, congratulations on your work that you're doing. Next is going to be Dr. Tina Stankovic, who's an MD and PhD, who's the chair of Stanford, and also has been very involved in multiple different areas of otology, She's been recognized as a leader in the field of treating hearing loss. And so appreciate all the work that you've done, Tina, in many different areas, but specifically, thank you for joining us today and talking about your work in perilymph sampling in humans. Let's see, Tina, are you on mute? We don't hear you yet.
There we go. Okay,
There we go.
can you hear me now?
Yes, we can hear you.
Thank you for the opportunity to present our work. And I would like to also gratefully acknowledge the funding from the government, several foundations, and grateful patients and families. To motivate the study of secreted molecules that may influence hearing, I'd like to start with a conundrum, and in particular, looking at vestibular schwannoma. So I'm showing a contrast-enhanced MRIs of two patients of the same age and same sex. And you can see that the patient on the left has a small intracanalicular vestibular schwannoma that caused dramatic drop in audiometric thresholds and even worse drop in word recognition scores, so that it's only 8%. And in contrast, on the right, you see a larger tumor with the same or similar intracanalicular extent, which caused much less hearing loss, and word recognition was maintained at normal 100%. So now if we look at many more tumors as opposed to looking at this anecdotal finding, and if we categorize these tumors into those that are purely intracanalicular versus extending a little bit into the cerebellopontine angle versus touching the cerebellopontine angle, or indenting the brainstem, then we can see when we analyze nearly 500 patients that there really isn't a correlation between tumor size plotted on the horizontal axis and the severity of tumor-induced hearing loss plotted on the vertical axis when quantified either in terms of pure tone average shown on the top or word recognition scores shown at the bottom. So, if these tumors do not cause hearing loss only by compressing the nearby auditory nerve, what is the mechanism? Over the last decade, we have provided evidence that these tumors secrete ototoxic and neurotoxic factors that can damage hearing. And we have also shown that pharmacologic and molecular blockade of these tumors can be therapeutic. So to then measure precisely these tumor-secreted molecules that may influence hearing, we are looking at liquid biopsy that could also guide future therapies. And the liquid biopsy includes not only collection of blood samples from these patients, but also collection of their inner ear fluid, namely perilymph. And to enable future routine liquid biopsy of the human inner ear, we first defined the proteome of human perilymph in both normal and pathology cases. And thank you, Idel and Heinrich, for referring to that work. We have also developed a microneedle device that allows reliable and controlled cochlear liquid biopsy, and it's a very simple device. It's basically a beveled microneedle with a mechanical step-off to prevent overinsertion, and it also has an electrical safety feature. And finally, we have demonstrated the feasibility of detecting molecular biomarkers of hearing loss in as little as half a microliter of inner ear fluid. So I will first start with a blood biopsy, so liquid biopsy of the blood in patients with vestibular schwannoma. And this is the largest cohort of patients studied in that regard, including over 100 patients. And we analyzed 67 different immune-related proteins and cytokines in the patient's blood compared to healthy controls. And the data are depicted where the vertical axis presents the concentration, and these are violin plots. And we focused on the cytokines that were detectable in the plasma of at least 75% of patients, making them candidate biomarkers. And we then zoomed in further only on those that I expressed at higher levels in the blood of patients with vestibular schwannoma, and these are the ones depicted in blue. We were happy to be able to validate these findings using a different cohort of patients from across the continent. And in particular, we validated 13 out of 14 of these candidate biomarkers. The next step was to define whether any one of them could really serve as a true biomarker. And to answer that question, we used receiver operating characteristic analysis, where we plot sensitivity on the vertical axis and 100 minus specificity on the horizontal axis. And then we can measure the area under the curve, and the greater the area, the better the biomarker. So when we combine 9 of these biomarkers together, they have an outstanding discriminatory ability to diagnose vestibular schwannoma compared to healthy controls simply by biopsying patients' blood. And you will see that one of these 9 biomarkers is tweak. And I will tell you a little more about it shortly. So in addition to collecting patients' blood, we can also collect tumor secretions. And the way we do that is when we remove these tumors during indicated surgeries, we can put them into culture media, and then we can collect tumor-conditioned media. And when analyzing the secretome of that fluid compared to the secretome of blood in these patients, there is a significant overlap. And I'll zoom in on one of these molecules in particular, namely tweak, that I just told you about on the previous slide. Tweak stands for TNF-related weak inducer of apoptosis, and we decided to zoom on it because we had previously shown that TNF is an ototoxic molecule secreted by vestibular schwannoma, where TNF stands for tumor necrosis factor. Moreover, we have shown that the higher the concentrations of TNF in tumor conditioned media, the worse the hearing when measured either in terms of pure tone average or word recognition scores. We can further divide tumors into high or low TNF secretors, and those that are high TNF secretors are more often associated with non-serviceable hearing, which is defined as having word recognition scores of worse than 50% and pure tone average greater than 50 decibel. And when we look at that TNF high secretor group, we see that there is a correlation with tweak, meaning the higher the concentration of tweak, the worse the hearing. So that's in the blood and in the tumor secretions. How about perilymph? So to answer that question, we first zoomed in on mice, where we developed an anatomically accurate mouse model of vestibular schwannoma that entails stereotactic placement of tumor cells into the cerebellopontine angle. The beauty of our model is that we can transfect these tumor cells with Gaussian luciferase, which is a secreted molecule that we can measure in all bodily fluids. And in particular, if we measured it in the blood, it gives us a very accurate representation of tumor size. So the first step was to actually measure the concentration of Gaussian luciferase in mouse's perilymph versus blood. And we see that it's concentrated in the perilymph at higher levels than in the blood. This is highly significant because this is the first direct demonstration that tumor-secreted molecules can reach the inner ear. So the next step was to look at TNF-alpha and tweak in these mice, and we find that, again, both of these molecules are secreted or found at much higher levels in the perilymph than blood, and at much higher levels in the ear ipsilateral to the tumor compared to the control contralateral ear, consistent with these mice having hearing loss on the side of the tumor. Well, how does this compare with human samples? To answer that question, we collected perilymph from patients with vestibular schwannoma and first found that both tweak and TNF are found at much higher levels in the perilymph of these patients than in their blood. Moreover, when we look at one patient at a time and plot their individual tweak and TNF levels, we find that patients with the highest concentrations of these molecules in vestibular schwannoma condition media also have the highest concentrations of these molecules in their blood and perilymph. So how are these two molecules related? To answer that question, we used auditory cell lines and treated them with tweak or TNF individually or in combination. And what we find is that these two molecules greatly amplify each other's ototoxicity because when combined at the physiologic concentrations that we measured in patients' blood and perilymph, they are much more toxic than either molecule provided alone. So in summary, what I have shown you is that vestibular schwannomas secrete ototoxic molecules and that liquid biopsy of perilymph and blood can not only be diagnostically useful, but could also be enabling future therapeutic options that are very targeted. Overall, our approach is consistent with Stanford Medicine's focus on precision health, which aims to predict, prevent, and cure precisely. Thank you.
Tina, that was fantastic. Thank you very much for, again, all the work you've done. And this is a great presentation. Really appreciate it. Moving forward, then, we're going to have Andrea Van Butis, who's the chair of the Department of Otolaryngology at Long Island Jewish Medical Center and North Shore University Hospital. She's the director of the Hearing and Balance and New York Head and Neck Institute. And she's been very involved in autoimmune inner ear disease and going to tell us about her work and how perilymph sampling influenced some of the work. So Andrea, thank you for coming on board. Dr. Van Butis.
Thank you so much for the invitation. They've been great talks so far. So anyway, I wanted to talk about perilymph sampling and how we used it to really change our research focus. So we study autoimmune inner ear disease, and for patients with autoimmune inner ear disease, 70% of them initially respond to corticosteroids. However, with repeated insults of dropped hearing and repeated use of steroids, only 14% of patients stay responsive to steroids. And unfortunately, that leaves us with a strategy of either hearing aids or cochlear implants. And so what we did is we took a look at patients with end-stage autoimmune inner ear disease, and specifically compare them to patients with long-standing stable sensorineural hearing loss that were undergoing cochlear implantation. And what I'd like to highlight about this is that the small cohort of patients that we looked at had various different systemic autoimmune diseases. So one had rheumatoid arthritis, one had type 1 diabetes, one had Hashimoto's thyroiditis. Additionally, all of the serologies were highly different between all of the patients. Nonetheless, we decided we're going to compare them. And so what we did at the time of cochlear implantation, we took a neonatal spinal catheter and harvested perilymph at the time of cochlear implantation and used it to specifically stimulate their peripheral blood immune cells. So we did not take a look at what the contents of that perilymph was, but rather asked, what did it do when it stimulated their own autologous peripheral blood immune cells, and compared it to the patients that are controls. What we also did is we vaccinated all of these patients 2 weeks prior to surgery with Pneumovax to give them an irrelevant antigen, and use that irrelevant antigen also to stimulate their peripheral blood immune cells as a comparator, so we could really have a robust comparison between untreated samples Pneumovax treated samples, and finally their own autologous Perilymph. And when we compare this by microarray, because at that time that was the standard way of doing this, only 10 different genes fell out, which was really interesting. And of them, I really want to point to the last two. The first is the interleukin-1 type 2 decoy receptor, which I'll talk about a lot more. Controls robustly make it, and the autoimmune patients did not. And the other one to just note is glutathione peroxidase. As you know, currently there are ongoing clinical trials with SPI 1005, which is a glutathione peroxidase mimic. And so we were excited about these results. And however, the question was, what do you do with it from there? And so what we did is we asked, well, what is a decoy receptor and how is it involved potentially? And so what we saw is that the decoy receptor is basically an immune sponge, and it is expressed in areas where we really have to regulate and control function. So it's preferentially expressed in the brain, in the eye, and perhaps now in the inner ear. And so what we did is then we asked, well, what regulates decoy receptor expression? And one of the things that regulate decoy receptor expression is corticosteroids. So then we asked, well, could we predict, based on decoy receptor expression, which patients with autoimmune inner ear disease respond to steroids and which don't? And what we did is we looked by quantitative real-time PCR in a cohort of 20 patients now prior to treatment and stimulated their peripheral blood immune cells with dexamethasone and very easily could identify and differentiate the patients that are steroid responsive versus the patients that were steroid resistant. Why was there such a large difference? Largely because the patients that are steroid responsive don't express any decoy receptor prior to stimulation with corticosteroids. And the patients that were steroid resistant had very high levels of decoy receptor expression. So that led us to work backwards. And what we found then is that the patients that were not corticosteroid responsive had very high levels of IL-1 beta in their plasma compared to corticosteroid responsive patients. And in fact, when we took those peripheral blood immune cells and stimulated with dexamethasone, saw very high levels of interleukin-1 beta release. Yet when we stimulated with anakinra, which is an IL-1 receptor antagonist, we saw a very nice blunting of that. That led us to a clinical trial. This is anakinra or Kineret. It is a IL-1 receptor antagonist. It's a daily subcutaneous injectable. And actually for our initial studies, we thought it was a great molecule to use simply because it washes out within 24 hours, and we really weren't sure what the effect of this drug would be. So our initial trial took patients that were steroid resistant for a period of 30 days where they received high-dose corticosteroids. They were enrolled into the anakinra trial. This was an open-label trial where they were treated for 84 days, and then they were followed for the remainder of the next 264 days. And what we found, and these are two representative samples, the upward arrows when they start anakinra, the downward arrows when they stop, we saw a very nice improvement in their pure tone average together with an improvement in word recognition scores, together with a reduction in their plasma interleukin-1 beta as shown in the green arrows. And this trial was prematurely halted by our data safety monitoring board because we had met our end target efficacy target earlier than anticipated. And the right panel just shows you what type of degree of response we were seeing in terms of both pure tone average and anakinra responders compared to non-responders. And on the far right is John Naparko's pivotal study on steroid responsiveness and autoimmune inner ear disease, which was the serial audiometry trial. And so the responses we were seeing were certainly comparable, if not better, with a cautionary tale of telling you that this was a very small study by comparison. And so it's to segue a little bit, and to look at a slightly related disease, as one of the things that we talk about is Meniere's disease. And we talk about sodium causing autoimmune disease. Historically, we think of Meniere's disease that it's the sodium potassium ATPase that is the molecule that really is responsible for the salt issue associated with Meniere's disease. But we now know in the autoimmune literature that sodium salt actually drives autoimmune diseases by increasing Th17 cells. In MS, we know that high salt diets actually cause exacerbation of disease. And so then we asked, well, does sodium really have any role in Meniere's disease? And indeed, what we see is that in Meniere's disease patients, excessive sodium salt releases interleukin-1 beta at both the transcriptional level and at the release level. And in fact, there's an alternate processing. Interestingly, diazide or triantropine hydrochlorothiazide actually reduces that release overall. And so that led us to a next current clinical trial where we're taking a look at the use of anakinra in both corticosteroid-resistant autoimmune inner ear disease and corticosteroid-resistant Meniere's disease. This trial is nearing completion. We passed our futility analysis, which was at 25% in. Could we see a difference between the blinded arms? There are 3 blinded arms, one that receives anakinra only for the same 84 days, one that receives anakinra for 42 days followed by placebo, or placebo for 42 days followed by anakinra. And so we're hoping this trial will be completed this year. However, throughout the course, and you can see this has been a long odyssey, we've had a number of patients that we have treated off-label because they didn't meet criteria for enrollment. And we've now, this paper is about to come out in otology, neurotology, but we had 3 cohorts that we studied, one that were ineligible for the anakinra trial, some of our original trial patients that could not maintain hearing off of anakinra, and then finally steroid-dependent patients that every time you tapered their steroid, they worsened. And what we found is that in the patients that did not meet eligibility, we saw an anakinra response rate of 46%, and in the steroid-dependent patients of 83%. So it seems as though we have an interesting marker that we originally identified with Perilymph. And as even though we didn't know the factors in the Perilymph, the very interesting thing is these are patients that underwent cochlear implantation, not as they immediately stopped steroids. So it was clearly some time afterwards from the time we harvested that Perilymph, suggesting that this was a durable marker. So with that, we also looked at these patients from this study that is about to be published and found that we could also predict that the patients that would respond to anakinra, if we took a look at the anakinra responsive patients from that cohort A, they dropped their IL-1 release in response to anakinra, yet the ones that did not respond to anakinra had no change. And so with that, I'm going to say thank you very much for the opportunity to discuss this with you today. Our studies are ongoing, and thank you.
Andrew, thank you very much. That was a wonderful overview. Thank you so much. So I think we'll work on putting some ideas of protocols together. We'll circulate with the group, see what people think. Is there anyone that wants to take a lead on these different things? Or if not, I mean, we'll move it forward, but also happy to. One of the goals of the Hearing Science Accelerator is to find young investigators that have a lot of time on their hand, more than we do, that would want to take charge and put together a grant proposal with that we could all help them with. So if you have some youngsters that are in your academic departments that might be interested in this, let us know so we can maybe groom somebody to help with all that. So to the speakers, great job. Thank you. And again, thank you for everybody's comments. So appreciate it all. This is a lot of fun for me. I enjoy doing this, having time to get together and talk about this stuff.